All right. Thanks for that introduction. I probably could skip some of my intro slides. That was great. And it's really wonderful to be here, to be participating in this really innovative. I know it's been going on for a while, but it's still very innovative symposium, and to share my work with you all. Okay, so my lab studies evolution of microbiomes, and when I'm talking about evolution, I'm not talking about on very long time scales, although we are interested in that. Primarily, we're interested in short time scales, the evolution that's happening on you in your body, every single day. And we can really see that this is not just something theoretical. There's really, a vast potential for evolution in your microbiomes every single day. And we can see this because there are about 10 trillion bacteria in and on each of us, each of which has about 1 million nucleotides of DNA. And every time one of these nucleotides replicates, right, which is probably about once per day, there's about a 1 in a billion chance of creating a mutation. Okay, so if we do the simple math, we can see that every day in our microbiome, there are about 1 billion new mutations upon which selection can act. And so even if the vast majority of these mutations are deleterious or neutral, this still represents an enormous potential for evolution on us every day. To sort of think about this another way, if there's a species that's just 10% of the microbiome, every nucleotide in its genome is going to be mutated 100 times, every single day. Okay. And so in my lab, we sort of think about this evolution in sort of two ways. On one hand, we're interested in, just sort of understanding this evolution from a fundamental level, because evolutionary theory is still not really well developed for when ecology and evolution are working on the same timescales as we know is happening in microbiomes. And but really what I want to talk to you more about today is this front arrow, which is really using evolution as a tool to help us better understand microbiomes. And we need to really better still understand microbiomes at a mechanistic level, because despite the potential for probiotics that we know today for improving health, getting to this reality of improving health with probiotics or therapeutic microbes is difficult because of ecological problems, right? I could probably team up with a genetic engineer to design a strain of C. acnes, a strain of bacteria, let's say, that produces sunscreen in response to light, but getting that bacteria to engraft on my face is going to be a challenging problem. Why? Because most microbes don't really stick, right? So this is an example of a microbe taken from humans, given to humans, and after removal of probiotic administration, you can't detect it in half the people anymore. And so we don't know what the difference is. Oh, sorry, I've been talking and now I see that the slides are changing. Sorry. Can you guys see the slides changing?
I see the probiotic curve.
Oh, you do? Okay, so my social there was a note that someone said they can't see the slide changing. So they're good.
Yes.
You're good.
Okay, good.
Hi, everyone. We're back. There's still just this, I think, is an example of just how much we don't understand in the microbiome. And the lens that my lab takes is that that within person evolution is a really good tool for sort of understanding mechanism in the microbiome. By tracking the mutations that bacteria acquire during colonization, we can reconstruct past evolutionary history, both between people and within an individual person. And by looking at what genes are being mutated, and what pathways are changing in vivo, we can sort of get a bacteria's view of the selective landscape that it's facing. And so a question you might be asking yourself is, if we see this large potential for evolution and this tool that it can be used for, why hasn't it been studied more? And there's a couple different answers, but I think the largest one is a technical issue, which is that from metagenomics, short reads of DNA from a mixed sample, it's actually very hard to study within person evolution, because what you'd like to see are mutations that have emerged from on-person diversification and evolution. But distinguishing these mutations from those...
Tami, just a second. I think when you're clicking your slides, they're passing to you, but now they are moving.
So they're too slow.
Yes, for some reason. Could you just
Yes, I'm so sorry.
move with the arrow, move with the arrow to forward and backwards, just so that we can see if it's moving.
With the arrow on the bottom.
Just arrow forward and arrow.
Yeah, I've been clicking.
Backwards.
Are you seeing it?
Oh, okay. Now I can see them moving as they're moving.
But I think it's a little delayed. Sounds like I'm doing a click right now, and you saw the.
And I see lineage 1 and lineage 2 just appeared.
Oh, okay. So I think that there's a significant delay, and I wonder if it's because of how I shared things. So let me try to just share things differently. I shared things slightly differently last time when I was practicing. So it's better this one. Hopefully this should be better. Okay, so I click.
I see lineage 1, lineage 2. Colony
Okay,
sequencing.
so I'll know that there's a delay. I can move.
Okay, there is a little bit of a delay, but I think it's working. Now it seems to be working. Thank you. Sorry to interrupt.
No worries. Hi, everybody. So what we're trying to talk about is why metagenomics is so difficult for studying within-person evolution, and the problem is that we really want to identify variants that are coming from on-person evolution, and we want to distinguish these variants from those that are coming from pre-existing variation, from say 2 strains of the same species colonizing a person, or 2 strains of different species even sharing the same mobile element, and really distinguishing between these classes of mutations from metagenomics is just really hard. We love algorithm development. We build tools for metagenomics in my lab, but really getting a precise classification of these mutations is very hard. And then furthermore, if we are able to remove these mutations, it's hard to know which de novo mutations are really coming from the same cell. And so to get around this, in my lab, we use a technique that's really relatively old school of culturing, where we culture many colonies of the same cell, many colonies of the same species on a plate from a single sample. And because each colony is formed by a single cell, this is kind of like single cell sequencing. We then sequence the genomes of each of these isolates independently, perform evolutionary reconstruction, and focus on those mutations that happen at the tips of the tree. While this sounds maybe expensive, it's actually not. When you do this in high throughput, like we do in my lab, including sample prep and sequencing, it's less than $15 per isolate. But the downside, of course, is that you need to focus on a single species at a time and have some sort of media that's relatively enriching for your bacteria of interest. Using this approach in my postdoc with Eric Alm, we did find the first evidence that there really is adaptive evolution in the gut microbiome, even in really healthy people, stool donors for open biomes. So their stool is literally medicine. But despite this sort of healthy state, we see really strong evidence of adaptive evolution, where on the phylogenetic tree from one person, we can see that different parts of this within-person tree have similar mutations in the same gene. And this signature of parallelism is a strong indicator of adaptive evolution. Moreover, these same genes showed high levels of DNDS, or rather, which indicates a pressure to change the amino acid sequence relative to a neutral model, whereas other genes didn't. So this really told us that these genes were under adaptive evolution in these people. And the particular genes that it pointed out were genes involved in outer membrane synthesis and genes that are involved in the import of complex polysaccharides, telling us that these genes really need to change in vivo. One thing that was really cool from the studies, we really got this really interesting picture of dynamics, where from the colonies, we were able to infer that there was this person's population was founded by a single cell ancestor about 8 years prior to the start of the study. And then tracking this phylogeny with metagenomics from every other day, we were able to create this really beautiful picture which shows us really interesting dynamics where despite the fact, where despite the fact that there's really adaptive dynamics within one of these lineages, right, this purple genotype outcompetes its purple ancestor, this overall sublineage does not really compete much with this other sublineage. They really stay at a remarkable stable frequency despite these adaptive dynamics. And so this pointed us to a hypothesis that perhaps these organisms are doing different things in vivo. But because it's so hard to sample spatial structure in humans, it's hard to understand if this is actually functional diversification or if it's just neutral separation in space. And so in my lab, which opened about 5 years ago, we've been focusing on the skin microbiome, where we can really access these questions about space to resolve different hypotheses. We've been focusing largely, but not exclusively, on facial skin because it really is a very rich microbiome. The fluorescence that you're seeing in this image is actually fluorescence coming from porphyrins, which are molecules secreted by C. acnes, the dominant resident on the skin. And so the first story I want to tell you about today from my own lab, we asked why different strains of the same species are coexisting on the skin. Are they coexisting from adaptive forces or neutral forces? And it's just occurring to me that Erilyn did present this work years ago, I think last year at MVIF. So if you've already seen this, my apologies, but hopefully you'll enjoy the refresher. And so the skin really is a really nice tractable system, the facial skin, because we can sample it at many different spatial scales, including the level of individual pores. It's relatively low diversity, where most of the community really is one bacteria. And it's really stable over time. It's not just something we're sampling from the environment. People really have unique facial skin microbiome communities. When I say it's low diversity, it really is, right? Most of this is C. acnes. But it's not really as boring as that seems. There's actually many strains of C. acnes on each person's skin. Before I go further, I want to clarify that C. acnes is really considered a commensal, despite this scary name, and that it's anaerobic. It really likes to grow in the bottom of the pores where there's lower oxygen, although it can survive at higher oxygen tension. We collected samples from 17 healthy people using a toothpick to get at larger spatial scales and really just standard cosmetic tools to get the contents of individual pores. From each of these samples, we cultured and sequenced the genomes of a handful to dozens of isolates, and then performed whole genome sequencing and evolutionary reconstruction. Okay, so what do we see from all this data? Right? Well, the first thing we can see is that it's clear that people are colonized multiple independent times in the environment. We group isolates into these groupings called lineages, where isolates within a lineage are separated by less than 50 mutations across their whole genome and are likely to have come from a single colonization event on that person. And we can see that just this one subject has 6 different lineages from a C. acnes tree, including lineages that are actually very closely related and have very similar gene content. We find very similar patterns across other individuals where there's many different coexisting lineages. And I just want to point out before I go further that we have really uneven sampling of our subjects. And so for the most part, I'll focus on these subjects from which we've sampled very deeply, but we find similar trends on these other subjects as well. Okay, so what do we see? We see that these lineages aren't exclusive to different parts of the face, and we find, for most cases, really coexistence on these different body parts, suggesting that spatial, regional selection is maybe not be driving this coexistence. And similarly, we don't find a huge difference between what we find on the surface and what we find in the pores. But when we zoom into the level of individual pores, we do find something very interesting. So from subject A, if we were to pick 2 isolates at random from a single pore, given the distribution of lineages on this person, we'd expect them to be from the same lineage about half the time. But when we look really at the individual pore level, we find that they're from the same lineage almost like all the time, suggesting that pores really are dominated by single lineages. To get some idea of spatial resolution, we can see here from this pore strip that there were 3 pores within millimeters of each other, but each of them had a different lineage dominating that pore. So really these islands are these, so these pores are really these distinct islands with their own distinct genotypes. And this raises the possibility that perhaps each pore selects for a different lineage. And to understand this, what we can do is we can zoom in to the within lineage level, because if each pore is selecting from the environment, and so saying only isolates from this particular lineage can live here, then we might expect to find each pore to be colonized by isolates from all over the within lineage tree. But when we zoom in and we look at the level of individual pore, we find this dramatic segregation by pore, where isolates from the same pore really are almost identical across their whole genome and share mutations found not found in other pores. So what this tells us, right, is what we're seeing is that this within pore diversity is very low, despite there being lots of diversity within a lineage, suggesting that it's not just sifting from the environment, but rather that there's a single-celled ancestor within each pore that existed a relatively short time ago. And we see similar trends across other lineages and other subjects. And so this raises the possibility that perhaps selection is very strong, right? Perhaps each pore has its own unique selective pressures, and these mutations that we're seeing that are pore specific are reflecting those pressures. And if that were true, we'd expect to see rampant adaptive evolution. But that's not really what we see. What we see is very strong signatures of purifying selection and no evidence of adaptive evolution. We don't find any parallelism, and we don't find an enrichment of amino acid changing mutations. And so this really suggests that whatever's causing these bottlenecks must be a neutral force that's not adaptation. I want to mention that this is not something that's trivial, right? You can have millions of bacteria in a single pore, but yet they're almost identical. So we have these recent single-cell bottlenecks. What's causing them? Well, we've created a biophysical model that we think can explain this pretty well. We know that the bacteria grow faster at the bottom of the pore, where it's anaerobic and nutrient-rich. The sebum is being produced at the bottom of the pore. And we believe that there's flow from sebum produced at the bottom of the pore to the top. And so if we make a very simple biophysical model, what the result is that it's very hard for a cell to get to this preferred region. And by the time a second cell can manage to get to this preferred region, the first cell has taken up space and physically occluded new entrants from being able to come in. So we get this priority effect at the level of individual pore. And this priority effect we think is really exciting because it can help explain the coexistence at the face-wide level. Okay? So even if this purple bacteria is much slower at growing than this green bacteria, because they're each segregated into their own pore and have their own access to nutrients, they're going to be contributing to the surface population at relatively equivalent rates, slowing the competition between these genotypes. So what I've shown you is that genotypes are really spatially segregated across the skin. We do not think that this is an adaptive process, and that there's a priority effect at the level of individual pore that can help explain coexistence at the level of the face. And we think this is exciting because it's an example of skin anatomy shifting the balance away from adaptation and towards neutrality. Therapeutically, what this means is that if we wanted to design a C. acnes-based therapy, which people are actively doing right now, we shouldn't worry so much about finding the right genotype for the right person, which we might need to do in the gut, but instead, what we should do is really focus on removing the current population that's living there so that we can create open pores for these C. acnes to colonize. You might be asking yourself, why are these different strains? How do we get these strains in the first place? And we are actively studying this, and I'm just going to tell you one quick highlight from an ongoing study led by Jacob Baker in my lab, where we've been studying children and parents at a K-8 school in the Boston area, and we can find transmissions of C. acnes from mothers to children and from a family member to family member. One cool result from this ongoing study is that when lineages are transmitted from one person to another, it's not a monophyletic grouping. It seems that multiple cells are transmitted from person A to person B at the same time. And this gels nicely with this idea that pores are their own ecological units, because perhaps each cell is sort of landing and colonizing pores independently. So I just want to be clear that while we're proposing that spatial structure is limiting competition at the community level, we're not sure why DNDS is so low in C. acnes, why there's no evidence of adaptive evolution. It could be spatial structure that's limiting the efficacy of competition, but there could be other forces as well. And so, the environment in the gut might be much more diverse from person to person than the environment of one face to another face. In the skin, we don't think everything is neutral. And in fact, I want to share a quick story with you about one case where adaptive evolution is happening on the skin. And this is the context of Staphylococcus aureus, an opportunistic pathogen that often lives in our nares, that really on atopic dermatitis skin. And it's been known for a very long time that Staphylococcus aureus really grows to very high abundances on atopic dermatitis skin. It's not thought to necessarily initiate atopic dermatitis, also called eczema. Initiation of AD is really thought to come from a compromised skin barrier due to genetic defects or environmental damage. But this compromised skin barrier enables Staph aureus to grow. And Staph aureus, in return, secretes immune factors, punches holes in cells, and further compromises the skin barrier. And so you get this positive feedback cycle that's pretty hard to break. Our best treatments for eczema work on either arm of this feedback cycle. They either try to fix the skin barrier or lower the immune response. There's been previous work that's shown that perhaps different strains of Staph aureus contribute to disease severity. And what was shown by these authors was that strains from people with AD caused worse disease in a mouse model. But what was weird was that despite this sort of association with disease severity, there wasn't a single genotypic strain of Staph aureus that could really explain this difference. Each person here really has a different part of the phylogenetic tree of Staph aureus. And so to understand more about how Staph aureus diversity is generated, we performed a longitudinal study with my collaborators, Maria Teresa Garcia Romero in the Institute for National Pediatrics in Mexico, where we sampled from affected and unaffected body sites and sequenced up to 10 whole genomes from each body site over 5 different time points. What we found is that something really different than C. acnes, really people are dominated by a single strain of Staph aureus. And the Staph aureus that we find in our collection really reflects the global diversity of Staph aureus with no real association with AD. So again, each person here really has a single lineage throughout this 9-month time points. Although we occasionally do see lineage switching or the coexistence of minority lineages. Almost always we find the same lineage in the nares that we find on the skin. To understand evolution, we performed really careful work where we developed a patient-specific assembly for each person and identified de novo mutations in relation to this patient-specific assembly. And the first question we asked is, does this inflammatory environment of inflamed skin increase the mutation rate of Staph aureus? And for the subjects with enough cultured Staph aureus to really fit per person molecular clocks, we find values of the molecular clock that are very similar to Staph aureus molecular clocks from other contexts, including other infections and healthy skin. And so we don't think that AD itself is increasing the mutation rate in atopic dermatitis of Staph aureus. But we do see really interesting dynamics. And so this is a phylogeny of one person. And you can see here the scale bars is one mutation across the entire genome. And so when we color this tree by where isolates were taken from, we can see that each genotype is sort of found randomly distributed throughout the body, suggesting pretty rapid spreading of these genotypes. But when we color them by time, we see an interesting segregation here, where isolates collected at this last time point share mutations that we couldn't find at earlier time points. So this really presents this picture of generation of new mutations that then sweep the body and replace their ancestors within months. And this pattern is common across all the subjects for which we had enough data to draw these nice trees, nice evolvograms. And from the other subjects, we find evidence supportive of recent sweeps because the population within each individual is inferred to have a pretty recent single-celled common ancestor. So each of these subjects was either colonized within the past couple of years, or there was a selective sweep on that person within the last couple of years. To understand if this evolution is adaptive, we can look for signatures of adaptive evolution, and we in fact find some. So we find parallel evolution in this one subject, where we find the same gene, despite there being only about 100 mutations on this whole tree, the same gene is mutated 4 different times, including 2 stop mutations and 1 stop mutation that's associated with a sweep. And so we see within person parallelism, one of these mutations being associated with a rise in frequency in the same person, and across person parallelism, where mutations in a different subject, we found a mutation in a different subject in the same gene that was also associated with a sweep. Okay, so what's going on here? What is this gene, CAPD? This gene is really essential for the production of the capsular polysaccharide in the outer cell wall of these bacteria, and breaking it makes these naked. And that seems to be a weird thing because in bloodstream models of infection, acapsularity is shown to be sort of a detriment and a deficit. So we thought maybe something was wrong, and so we analyzed public data to see if we could find this trend in more than just our subjects. And so we downloaded isolates from studies where. We had downloaded genomes from studies where single isolates were collected from people with AD or without AD. And what we find is that from healthy people, you almost never find CAPD truncations. You almost always find the full-length gene. But in about 25% of people with AD, you do find CAPD truncations. And this is actually a very similar number to our study, because in our study, while we found 2 subjects that had isolates. In our study, we found 2 subjects that had CAPD mutations that swept their populations. Another 6 subjects had CAPD truncations at the initiation of the study. So a very similar number in our study. So what we see here is that there's an association of CAPD loss with AD, even though there's no strain necessarily associated with AD. So we don't know yet exactly why CAPD loss is adaptive. There are many potential possibilities. We know that antibodies can target the capsule polysaccharide. We know that when you don't have capsule when you don't have the capsule, the adhesins on the on the cell wall can better attach to the skin without having this big negatively charged molecule in the way. But one thing that's sort of maybe the simplest is that perhaps by not making this expensive polysaccharide, you can just grow faster. And we were able to confirm this in the lab that in just a single generation, there's a 1% growth advantage for the capsule negative strains, and this contributes to a much larger advantage during exponential growth. Okay, so what have I shown you so far? That Staph aureus lineages rapidly spread across the body in people with AD. This appears to be adaptive, with CAPD loss being common. And we don't yet know if this contributes to worse disease in humans. But regardless of whether or not, I mean, it means that if we are going to make any strategies to eradicate Staph aureus and AD, we need to make sure that they're also effective on these capsule negative mutants. While we weren't able to show that this cause worse disease in a mouse model, one can still imagine that perhaps this adaptation is important for this positive feedback cycle. Because if we believe this model of a positive feedback cycle, we can imagine a second positive feedback cycle. Whereas Staph aureus is growing on a person, it's acquiring de novo mutations that better live on that person's, that help them better live on that person's skin in the context of that person's treatment, whatever it might be. And this might contribute to faster growth on that person's skin. This would then further exacerbate this arrow of Staph aureus contributing to the compromised skin barrier and make this positive feedback cycle even harder to break. And so we kind of like this idea that sort of bacterial evolution might be exacerbating complex diseases like atopic dermatitis and perhaps others, but this is still a model and just a hypothesis. So we don't have a good sense of the time. And so I think we're running low on time. So I think I just want to mention just a couple quick things here, which is there's some really interesting questions that come out of this study, which is why is there this association at the gene level but not at the global level? And we don't really know, but it's something we're really interested in trying to understand. Is are there priority effects? Are there host-specific selective pressures that are preventing the strain that adapts to me from spreading to my friends with atopic dermatitis? We don't know. And there's another really interesting question, which is, shouldn't these things that are breaking genes be deleterious in the long term, right? Wouldn't you want to turn a gene down in a more subtle way? And the answer is, well, maybe these mutations that break a gene cannot spread, but maybe when you have loss of function mutations, they're not necessarily dead ends, because perhaps given the very, very large population sizes of bacteria, it's actually not very hard for the same cell to mutate back and repair that loss of function gene and that gene. And for the sake of time, I think I'm going to end here, but I just wanted to advertise that Paul Torillo, my lab, has recently put a paper on bioRxiv exploring the potential of reversions in bacterial evolution in the microbiome, and we think that it can help resolve this long-standing paradox where the same genes that we find to be adaptive within an individual level show signatures of purifying selection when you look at longer timescales. And so before I end, I just want to say that we really love de novo mutations. We think that it's possible that de novo mutations, because they've been really hard to study, it may just be underappreciated, and that their functional role may be important in microbiomes. But even when they're not, even when these mutations are mostly neutral, we can use these mutations to help us understand about microbiome formation and mechanism. in healthy humans. This work was really very collaborative. I had really two amazing postdocs leading this work. Felix is now a group leader at the Max Planck in Berlin, and you know, all of our co-authors and lab members have really been so essential to getting this work done. I'd love to take your questions, both now and in the Pacific time.